Dear students, we will continue our previous lectures. Uh, Last time we have seen how the horses were developed. Now here the chapter is going to end. Uh, the questions, multiple choice questions will be there in the examinations, and they were given at the end of chapter. And these are the highlighted answers were always given for your betterment. So the remains or impressions of animal and plants have been found in rocks, mud, snow, or in soft sediments are called as fossils. Likewise, you can find all the answers. If you have any difficulty, you can communicate me. Meanwhile, about 16 objectives were given. You can prepare more than these and be ready yourself for the examinations. Now we will continue our lectures in unit number 4. So in unit number 4, the process of evolutionary change was studied. So we will study was given. So we will study it as a organic variations, isolating mechanisms, natural selection, example industrial mechanism, types of natural selection, directional stabilizing, disruptive, these are the natural selection methods, and artificial selection. So organic variations, isolating mechanisms. So we will start first topic, organic variation. In that isolating mechanisms, natural selection, example industrial mechanism. Types of natural selection are directional, stabilizing, and disruptive, and artificial selection. This was about the topic. Now, in organic evolution, the concept of organic variation holds that all the varied kinds of animals and plants which are not known have developed out of earlier types of completely natural, gradual, but continuous changes during the passage of time is nothing but the organic variation. But according to G.L. Stebbins in 1976, organic evolution is a series of partial or complete and irreversible transformations of the genetic composition of populations based principally upon altered interaction with their environment. It consists of chiefly of adaptive radiations into new environment, adjustment to environmental changes that takes place in a particular habitat, and the origin of new ways for exploiting existing habitats. And these adaptive changes occasionally gives rise to greater complexity of developmental patterns of physiological reactions and interactions between population and their environment. So this is nothing but the organic evolution. So in that we can study the series of partial or complete and irreversible transformations of the genetic composition, because the genetic elements are playing a key role in the evolution or development of the animals in an environment. The product of organic evolution is the origin of species or evolution of old species into new species. For example, during organic evolution under certain environmental stresses, certain autotrophic, heterotrophic, anaerobic, and aerobic prokaryotic cells of kingdom Monera started symbiotic relationship and evolved into eukaryotic plant and animal cells to form kingdom Protista. Later on, kingdom Protista evolved and gave origin to three kingdoms: plantae, fungi, and animalia. So, in evolutionary terms, differences between closely related organisms are termed as variations. Listen here, the evolutionary terms, differences between closely related organisms are termed as variations. And thus, the differences between these individuals of some species or some races can be called variations, and not differences between two genera or families or classes, only differences between the species. So only heritable changes have evolutionary significance, and they are considered for evolutionary studies. So kinds of variation includes the group variations and individual variations. In group variations, example, the man in Africa, they were black, while European population is white. So these were the variations. And individual variations includes the important taxonomic studies, in which we can study the population in a specific taxon. So variation can also be classified in the following categories based on the type of characters that we consider for the evolution. Is the meristic variations. These are variations that can be counted in numbers. For example, man has 12 pairs of ribs, but if some individual has 13 pairs of ribs, it will be called a meristic variation. Similarly, some people possess 6 fingers instead of 5, and starfish may have 6 arms instead of usual 5. So this is the meristic variation. We cannot call it as a new species or new, but this is the meristic variation. So quantitative variation that can be changed or measured in size or weight, tall versus dwarf, heavy versus light. In this way we can quantitative variation consider. So qualitative variation includes the characters, because some individual depict specific characters and presence or absence of spots, hairs, colors, stripes, feathers, etc. These are the qualitative variations. So based on continuity of the character, the variations can be classified in the two categories. One is continuous variation, also called as clinical variation or minus plus variation. So in that we can see the fluctuations with intermediate stages. Population of some individuals are larger, some smaller, and some intermediate in Indian population. Then this continuous variation includes the variation deviate greatly from the average individuals and major mutations and disruptive selections produced in these individuals. For example, white tiger, then hornless calf, etc. Based on inheritance, so variation can be classified into the following two types: somatic variation and genetic variation. So here the somatic changes are occurred as per the changes in the environment, and genetic variations they were produced in the phenotypic changes in the population. Some variations not classified in above mentioned ways, but they are of different. First, age variations. When young ones differ from the adult distinctly, as caterpillars differ from the adult butterfly or tadpole differ from the frog, it is called age variations. So young ones are looking like different and adults are looking very different. So these are the age variations. Like seasonal variations, commonly found in insects and butterflies. Then habitat variations, there are some sedentary, some sponges, oysters, as well as in plants variations. Some are mobile, some are free living. Example, chameleon can change its body color according to the habitat. This is called habitat variation. Then caste in social insects. So division of labor in social insects, example termites or honeybees, ants and wasps, creates castes which have specialized organs to carry out a particular job in the colony. For example, the worker bee has the pocket, so collect for collection of pollen and nectar. So it is called as pollen basket. So polymorphism. So there are different types of individuals occur in a single interbreeding population of a non-social animal called as polymorphism. This is found in butterflies and beetles very well. So sources of variation and somatic as well as genetic variations can be produced in the following ways: environmental factors, heat, cold, drought, food. These also drastically changes the variations in the animal. Second, endocrine glands. Variation in the endocrine glands is very important, and it's a functioning like pituitary, adrenal, etc. produces variations as per the changes in the somatic cells. Third, blending inheritance. The Darwin laid emphasis on the blending inheritances as a major source of variation in animals. In that, the characters segregate in the subsequent generations as per the law of Mendel, and the changes occurs. So mutations are the sudden changes that occur in the genetic setup and can change in their codon setting, either in the mRNA or its amino acid sequences, and causes changes. So these are the drastic changes. Adenine in the codon DNA make drastic changes whereas UUG is will not synthesize the different amino acids in some as the mRNA codes it for the process of formation of protein synthesis. So CTT coding for glutamic acid is also changed to CAT. In this way, the mutations can changes different setup of the DNA and its polypeptide chain. So role of mutation in a natural selection plays a key role in the changes because the formation of multiple alleles shows mutation. In diploid mutations, mostly recessive mutation occurs. The characters are not expressing very well. In haploid organisms, they affect the characters immediately. Then recombination of mutant genes takes place during the crossing over because this is the natural phenomena. When crossing over is over completed, then they again shows the changed characters after linkage. They uh, exchange the characters and forms a new one. So in this way, the new changes occurs in the animals due to mutation in the natural selection. Then in chromosomal aberrations like deletion, duplication, inversion, translocation, these are the different activities occurs after occurs after linkages in crossing over, and in these processes some genes may left behind or um, broken or then lost. So these are the changes. First, deletion. In that chromosome number 21 causes deletion of that particular segment, shows granulocytic leukemia in the animal, which is located on one arm of fifth chromosome, causes cry-do-chat syndrome in children. In Drosophila, deletion of in X chromosome causes notched wings. The wings shows notches. Duplication. When gene is represented more than one, it is depleted from one chromosome and get attached to the another one. Example, in Drosophila arises due to duplication. Bar I is the cause of this duplication. Then inversion, in a piece of chromosome breaks and joins at the same place after rotation. This is called as inversion, and this may cause changes in the position as well as in the genetic setup. Translocation involves transfer of one gene block from one linkage group to another non-homologous chromosomes. Then most of the translocations do not produce any abnormality in carriers as deletion, and 50% of these offspring carry heterozygous and will grow grossly normal. Then in aneuploidy, when the entire chromosome is either lost or duplicated, in Drosophila, the gene for white eye is X chromosome. When two X chromosomes fail to separate during oogenesis, female offspring with three X chromosomes will be super females which have low viability and are sterile, while some eggs will have no X chromosome at all. In man, non-disjunction shows XXX, generally called as super females, and XXY, generally called as the man with Klinefelter syndrome. And X0 means one is lost here, causes Turner syndrome, while Y0 is non-viable, and Down syndrome or Mongolism is caused due to these genetic changes in aneuploidy. Whereas polyploidy, here duplication of entire haploid set of chromosomes due to abnormal mitosis or meiosis, leading to triploid or tetraploid organism. Three or four gametes were produced, and these changes obtained from the seeds of Colchicum in polyploidy. This is the best example in the biosciences to obtain better fruits and flowers. So in polyploidy, the polyploidy is not mostly successful in animals, but it is mostly successful in plants. also found in streams, isopod, moss, beetles, etc. Hybridization. When isolating mechanisms between two species breaks down and they produce offspring, such offsprings are normally sterile, but sometimes they can backcross with the parents to produce second generation of individuals. For example, Raphanobrossica is produced by crossing Raphanus means radish and Brassica means cabbage, and both have 18 number of chromosomes. So hybridization is a common mechanism that produces variation in the invertebrates, particularly in insects. So next isolating mechanism that we will see in the next period. So meanwhile we will have a break here.